Students, today we are going to study the enzyme and its classification as per IUB and MB classification. Enzymes are the proteins that are used by the cell to speed up and regulate the metabolic reactions. Enzymes are biological catalysts that remain unchanged by the reaction. Enzymes are the proteins which catalyze the biochemical reactions without being consumed. Enzymes are the proteins which catalyze the biochemical reaction without being consumed. Enzymes are water soluble, they are produced by the living cell but can act independently of the cell. They are specific in action. Majority of the enzymes are protein with catalytic capability crucial to perform the different processes. There are some types of the enzymes. Exoenzymes, they are also called as extracellular enzymes. The enzymes which are secreted outside the cell are known as exoenzyme. They decompose the complex organic matter like the proteins, fats, cellulose. Examples of exoenzymes are proteases, lipids. Endoenzyme, second type of the enzyme is endoenzyme. They are also called as intracellular enzyme. The enzymes which are present inside the cell are called endoenzyme. Example are synthetase and phosphorylase. Third type of enzyme is constitutive enzymes. These enzymes are produced in the absence of substrate. Example is enzyme of glycolytic series. Induced enzymes, fourth type is induced enzyme. These enzymes are only produced in the presence of substrate. See, in the presence of substrate, induced enzyme, and in absence of substrate, it is a constitutive enzyme. Example of induced enzyme is hepatic microsomal enzymes. And last one type is zymogens or proenzymes. They are produced naturally in an inactive form which can be activated when required. Enzymes like pepsin are created in the form of pepsinogen, an inactive form. Pepsinogen is activated when the chip cell release it into a HCl, which partially activates it. Next are the properties of the enzymes. All the enzymes are protein in nature. Enzymes have extraordinary catalytic power and are active even in very small quantities. Enzymes are very sensitive to heat and temperature. Enzymes are very specific to their pH requirement. The substance on which the enzyme acts, it is called a substrate. By the action of the enzyme, substrate is converted into product. The interrelation between the substrate enzyme and the product is enzyme plus substrate, it gives the product. Most enzymes catalyzed reactions are reversible. Each enzyme possesses a tertiary structure and characteristic conformation. The functional unit of the enzyme is known as a holoenzyme, which is often made up of apoenzyme, the protein part, and a coenzyme, non-protein part or prosthetic group. Holoenzyme, which gives by apoenzyme and coenzyme. And last property of the enzyme is specificity of the enzyme. Enzymes are highly specific in their actions. Their specificity lies in the fact that they may act on one specific type of substrate, on structurally similar compound, on the specific optical isomer of the compound, on the specific geometrical isomers. These are absolute specificity, relative specificity, group specificity, bond specificity, optical specificity, and geometrical specificity. Next is the classification of enzyme. In the IUB system, International Union of Biochemists and Molecular Biology MB, IUB MB system, each enzyme has a name and unique identification number. The chemical reaction catalyzed is the specific property that distinguishes one enzyme from another. According to International Union of Biochemists, the enzymes are divided into 6 functional classes. These are classified based on the type of reaction in which they are used to catalyze. The 6 kinds of enzymes are hydrolase, oxidoreductase, transferase, hydrolase, lysase, isomerase, ligase. Except the enzymes tylene, pepsin, trypsin, ellipsin, enzymes are usually named by adding suffix ase to the name of the enzyme. Example, esterase acts on ester. Features of this classification of enzymes. All the enzymes have classified into 6 major classes. Each class is again divided into subclass and sub-subclass. In this classification, the enzymes are given a 4-digit unique number called as EC number. EC number is enzyme commission number. The 6 major classes of enzyme are as follows. First one is oxidoreductase EC01. These are the enzymes which brings about the biological oxidation and reduction and are linked to respiration and fermentation processes. Example are dehydrogenase, oxidase, hydroxyperoxidase, oxygenase, hydroxylase. Second one classification is transferase EC2. These are the enzymes which are responsible for transfer of functional group from one substrate to another. This is a transfer of functional group. That's why it is a transferase. Examples are transaminase, transphosphorylase, transglycosidase. Third one is a hydroxylase, EC3. These are the enzymes which brings about the hydrolysis or condensation of substrate by addition or removal of water. Example is esterase, phosphatase, glycosidase, peptidase, etc. Fourth classification EC4 is lysase. These enzymes catalyze the removal of group from the larger substrate by the mechanism other than hydrolysis, leaving the double bond. Example is aldolase, decarboxylase and dehydratase. Fifth classification isomerase EC5. The enzyme catalyzes isomerization, that is interconversion of the optical, geometrical or positional isomers. Example is alanine racemase, retinase isomerase, glucose phosphatase isomerase, epimerase. And last one classification is ligase. These are the enzymes which are responsible for joining or linking or synthesizing two components together with utilization of ATP, forming a C5 bond, CN bond, CC bonds. These are also called as synthetase. Example, succinate thiokinase, peptide synthetase, acetyl coenzyme, carboxylase and DNA ligase. Next lecture we will see mechanism and inhibitors of enzymes, that is enzyme mechanism and enzyme inhibitors. Thank you students.